chat if you have any questions to ask the instructors today throughout the duration of the presentation. While one is presenting, the other will keep an eye on that Q&A box and answer your questions in real time. Hopefully we'll be able to get to all of them and have enough time at the end for a little bit of a Q&A as well. Without further ado, I'm going to pass it off to Mary Ann.
Thank you. And hello, everyone. I am so glad to be here today. I'll say good afternoon for our East Coast people and good morning for West Coast and good day to everyone in between there. I'm really happy to be here and finally have an updated st91 for all of you. And we're going to go through some of that history in a few slides, but know that the new st91 was released as of the beginning of this month, and that's why we're having this training now. So as MJ said, my name is Mary Ann Drosnock, and I'm the Director of Clinical Affairs at Healthmark Industries. I've been in the role of education with Healthmark for almost 7 years now. Time sure does fly, and I love to present to people about infection prevention topics related to medical device reprocessing, specifically when it comes to flexible endoscopes. In addition to my role at Healthmark, I also am the former co-chair of Working Group 84, which is responsible for writing this st91 document, and TIR 99, which we'll hear more about at the end of the program. And I had been in that role since 2013, since its inception and idea, coming back from 2012 and officially starting in 2013 through late 2021, and with the approval from the working group and from the standards board then of the new st91, my term has officially ended and I'll be passing off that baton. So that's enough about me. How about you, Garland?
Hello. I echo the sentiments of Mary Ann about how happy we are that this document finally is able to reach everyone that needs it. For me, Garland Ray Grisby, I'm the endoscope service manager for East Bay for Kaiser Permanente. I have been with Kaiser for 18 years with the focus of endoscope processing, and I've been the endoscope service manager for 13 years. We've seen a lot of changes, and we were happy when we first saw this document in 2015. I've been a member of this Working Group 84 since 2014, and have been the co-chair with Mary Ann. She's been an excellent teacher and mentor. This is my second term, and we're happy to give you these updates in brief so that you have a better understanding of just what to look for with this new document.
Thank you, Garland. And the format is Garland and I are going to go back and forth with a set of slides, and while one of us is speaking, the other will be monitoring the question and answer tab to try to address those directly. It might be that we announce those questions, or we might answer you personally. Just depends on how the timing is going. And if we have time at the end, we will go through an open Q&A session. If not, I would be happy to answer questions, and you'll see our contact information on the last slide if you want to reach out to us personally with any questions. The other thing I'd like to ask is, Garland and I are trying to put together some additional topics for aami webinars. So if there's something that strikes your fancy during the program, please put that in the Q&A or in the chat or something like that, so we get some ideas of what to focus on next time for more pointed training on specific topics related to endoscope processing and how that falls within st91. So again, not only your questions, but if there's something specific you want us to focus on for a subsequent training session, please do. So with that, let's go ahead and get started. We do have 2 hours today. We have 60 slides to get to, so we should be able to get through everything with no problem. But many of you know that I can talk all day, and Garland's not too far behind. So let's see what we can do today. All right, let's advance these slides here. So as we said before, as disclosure, I'm an employee of Healthmark Industries, which manufactures and distributes medical products within the infection prevention, endoscope processing, and sterile processing arena. And Kaiser is an employee, Garland is an employee of Kaiser. So we are going through exact wording from aami and summaries of what's said within the document. If we do share our opinions as we go along, remember that those are our personal opinions, and you should always look at the instructions for use for your device or any other piece of equipment that you're using. So please keep that in mind as we go through. Our objectives for today are very simple. We kept it that way. We want to go through key differences, highlighting what they are between the original 2015 version and the new 2021 update. And by doing so, we will identify what are best practices related to the processing of flexible and semi-rigid scopes as is found within st91. Before we get started, I usually do speak to this slide in my presentations, so I thought it worth highlighting today just as a reminder where we fall as a standards organization for aami, because you do have this hierarchy. At the top of that list is regulations. Regulations are rules or mandates that come out from a governmental authority. You'll see these related to OSHA, regulations within st91, and those are captured by specific words, which we'll talk about in the next slide. But really, those are your top tier mandatory items that you'll find within the standard and outside of the standard too. Then as far as the standards organization fall, these are things like ANSI, aami. We have our national standards. Although they are considered voluntary for a facility to follow, they do become mandatory if your facility or your department states compliance with aami standards. Then it's expected that you do follow them, and you can be surveyed and inspected to those standards then. So we do, within st91 and other standards like st79, we have requirements in there and specifications that by following the standard, you ensure that you have a consistent process in place. Below that, then, we have guidelines such as those from professional societies, recommended practices, aami documents such as technical information reports, which are also called TIRs. These are also considered voluntary. But again, think about what you're claiming compliance to, and then you are expected to follow that guideline. So, and those are usually more specific information on a certain topic, like TIR 99, which will be related to probes and dilators processing, and very specific within endoscopes. It might be SGNA for GI scopes. So we see a narrower focus, whereas our standards really are all for, as far as st91 goes, all types of flexible scopes in all settings. So we have a much larger fit for that guideline. As I alluded to, words have meaning within aami and other standards. And it's very important that you understand what those words are, because that will help you with your interpretation of the document. And I always say, get the standard, start digesting it, see where you're at in line with your current practices, and start an implementation strategy then for getting to compliance with st91, because a lot of us aren't compliant with st91 right now, and we do want to get there. We have tons of evidence that we incorporated into the new standard to help give you the support that you need to make changes within your facility. So in order to go through the document, you have to know these words, and they're found at the beginning of every standard, st91 included, if you have a question about what they are. So the highest tier is must, and a must is those governmental regulations that we talked about. It means it is mandatory by that governmental agency. It's unavoidable. So these are things that are captured within the document related to bloodborne pathogens, training, transportation of contaminated medical devices, and these are captured by the word must within the standard. So if you see the word must, that means that came from a higher authority than just the standard. You have to follow that whether or not you are claiming compliance to st91. The next tier down then is a shall. And a shall means a requirement of the standard. So if you say you're compliant with st91, then that is something that you will have to do to remain compliant with it. So a requirement of the document is a shall. The next tier down then is a should. And a should is a strong recommendation. It's based on evidence that we've included in the document. It means you really should be doing something on that topic. There may be another way to do it other than what we say, but we can't really think of it as just wishy-washy when it comes to a should. There's enough evidence to recommend that you do something on that topic. Then we have a may. May gives permission. It means you have that permission to do something as part of the document as it relates to that topic itself. A can then is below that, just means something is possible or you may have the capability to do it. So again, use these words to really help narrow your focus as to, oh, these are a must. If I'm not doing that, that's my first thing that I've got to do. Or if it's a shall and I want to be compliant, then I need to put that high on my priority list. So I hope that's helpful. And again, found in the beginning of each of the standards that you're looking at. So let's dive into st91. I first thought it would be good to take a step back and see where we came from. As I said at the beginning with the introduction, we started kicking this idea around of a document related to endoscope processing back in 2012. And on that group started with Nancy Chobin. And quickly, we discussed that really this wasn't a technical information report, which is what we thought we should have at the beginning, so we could fast track something and get some guidance out there. But quickly we realized we needed more, and soon. So we moved that thought of a TIR into a full standard, and we started that in 2013. And it initially kicked that off, and we had a document published by 2015, which in the world of standards is pretty fast, considering we started updating the 2015 version once it was released, and here we are. It's got a 2021 date on it. So you'll see it took a lot longer to get through the second time with all the changes that we put in. But 2015 was the original version, and that's what was effective until this new document came out, or was released earlier this month. But what remains the same with st91 is the scope. This document, what's so great about it is it's applicable to all flexible endoscopes and semi-rigid endoscopes in all healthcare settings. So it holds everybody to the same standard of care. It doesn't matter whether you're processing your endoscopes in an endo suite, an endo department in a hospital, or they're going to sterile processing, or they're being transported to an off-site location, or they're in an ambulatory care setting or a physician's office. It holds everyone accountable to the same standard of care. And I think that's major, because we often see differences in what's being done in practice in your physician's office versus the ambulatory care center versus a hospital, or versus sterile processing or the bronch lab or whatever it is. But we do highly recommend within st91 standardization of practice across your different settings. So your first bit of homework is going to be figuring out where are my endoscopes being processed? What departments have scopes? How are they doing it? Don't forget about those off-site locations that are cleaning their scopes and processing them, maybe in ways that you didn't know that they were doing. So take a look at that. Strive for standardization. It really is a great aspect of improving patient care. We kept the same exclusions also. We don't deal with probes, dilators, manometry. Those will, at least the probes and dilators, will be covered by TIR 99 when that comes out. Hopefully it won't be too long, but who knows, right? And then our rigid scopes are also excluded, because those are covered within the scope of st91, because you have rigid stainless steel devices. What's also great about st91 is it covers all steps of processing. So from immediately after the patient procedure, when that is starting our point of use treatment, or pre-cleaning as we used to call it, back through reuse on the next patient. You get everything for all scopes and all settings within one document, leading us to standardizations. And that's what I love about st91. You can tell I get really excited about it. Sorry. This has been my baby from the very beginning. So I'm just so happy to be here telling you about it today. Now, as I said, st91, the new one, is available, and it has a 2021 date on it, which I know has confused a lot of people, myself included at first. But the reason it has a 2021 date is it was approved by the Working Group 84 late in 2021, and it was approved by the aami Standards Board in 2021. So that's why it's got that date on it. Although, it went through copy editing and everything like that for a while, and was finally published in 22. So on March 3rd, 2022, we did see this document officially be published. Again, so excited about it and happy to be here to share that information with you. So with that, we're going to start into what's new within the document. And I do see some chats and Q&A coming in, so we'll be looking at that as we go through. But let's start with what got us here, right? We've all heard of outbreaks related to flexible endoscopes, and that continues, unfortunately. Even though we hear less about it in the news media, we have seen multiple published reports of lapses within endoscope processing world that have led to tens of thousands of patients being exposed around the world to organisms and cross-contamination and infections. This continues. So we know that we needed to really get all the evidence together that we could and put out a much more stringent document so that we can help alleviate or reduce those risks related to patient infections and cross-contamination. So what we did this time is we did, like I said, a really good literature search on all of these topics, and included things like the FDA database reports from the MAUD database that are a requirement for facilities, healthcare facilities and manufacturers to make reports in there about major adverse events like serious injury, illness, or death. And there are many in there, unfortunately, related to endoscopes, whether it be failures in processing, repair issues where a scope that was in a state of disrepair was used on a patient and they were injured, citations related to cross-contamination of endoscopes leading to patient infections or unfortunately deaths that still occur, and other major issues. So it continues to be a problem, and that's why we started immediately updating st91 again, because we're learning so much so fast right now. There's a lot of research going on in the area. We see a lot more journal articles, peer-reviewed journal articles being devoted to endoscope processing issues and really trying to raise that bar as far as risks related to patients, lowering that, and making sure that we're building quality into the process. And that's really what we focused on in st91. Here's the steps that you need to engineer into your process to raise that bar and make sure we're protecting our patients. Because if not, we do have infections, unfortunately, that can occur. Often those are MDROs, as we call them, multiple drug-resistant organisms, bacterial infections, or superbugs, as they were called in articles. So we know that antibiotic resistance is a problem, and if we give these infections to patients, sometimes we can't get rid of them, and they can lead to serious, significant disease and even death. So we know we have this issue with endoscopes. What can we do? We can build quality in, as I said, we can institute the guidelines for as far as st91 goes, put those requirements of the shalls in the document, get those all in place, incorporate the recommendations that are captured as shoulds. But we still have difficult instruments to process. They have multiple channels. They have dead-end lumens, right-hand turns, depending on their configuration. We can have accessory channels that will never get brushed. It's impossible to do that. So we continue to struggle with those issues of the difficulty in processing them. We'll see that the manuals for the instructions for how to process these scopes are a book in length with hundreds of steps every time that endoscope is processed. So it's very difficult to achieve those cleaning instructions on an every time cycle. And we know these are significantly contaminated devices. I always say the most highly contaminated devices that you'll ever lay your hands on for processing. So we know we have high soil levels, difficult instructions with multiple steps, and just really highly engineered devices that are difficult, inherently difficult to clean. So that leads to poorly processed, poorly cleaned devices if it's not done right all the time and implementing the quality standards that we have in st91. It can lead to failed cycles for processing, and unfortunately that can lead to risks and infections in our patients. So with that, Garland, I'm going to turn it over to you.
All righty. So there was just a few questions in the chat that was answered. MJ answered a few. There were some about if the slides will be available and if the recording will be available. MJ answered that. There was a couple of questions about the TIR and when that will be out. I answered that. So there's a slide that discusses that as well. Hopefully, we hope to have that out by the end of this year or early 2023. And then there was also a question about rigid scopes and where that information was found. I answered that, so you can find that information in st91. So moving on to this slide here. Thanks, Marianne. Marianne mentioned, you know, the scopes that we touch, they're highly soiled, difficult to clean, and that they are. I recall when I first stepped in the field and saw my very first scope, it sat there for 48 hours. No one in the department wanted to touch it. I wanted to know why, and they said because it's hard to clean. I asked about the issue of biofilms and the soils building up, being that it sat there dirty. And this is why we're here today, because of, like Marianne said, just lack of cleaning, lack of understanding of how it's supposed to be cleaned, and the difficulty of the design, especially with the older model scopes. So with this 91 update, we updated and expanded the definitions section. We talked about high-risk scopes. Before, some folks would say, well, only certain types of scopes are high risk, those that have multiple channels. But what we found that there's been endoscopes associated with infections, some that we may feel are simple, such as cystoscopes or ureteroscopes. Those have been found and have been included in some of these outbreaks. So now we have bronchoscopes, cystoscopes, duodenoscopes, linear ultrasound scopes, EBIS scopes. All of those have been found as of lately through studies and data collection that, hey, you know what? These scopes aren't as simple to process as we thought, let's say a year or 3 years ago or 4 years ago. There's a few slides later that'll talk about the MOD database, where you can find some of this information on some of these reports that are out and how these scopes have been deemed to be high risk. So again, as Marianne mentioned about what scopes are being processed and where they're being processed at, this is a good time to go back to your facility and find out what scopes are actually being processed and see how they're being processed to determine in your facility what scopes are high risk and what you need to do about them. Next slide. So now here we have that word that should. So as far as the design or the processing area, some folks are stuck in old buildings, and it's difficult to design to the current way that things should be or the way that we should have it. Now, again, we had a conversation about this document and how this is actually helpful for folks in facilities, because once this document is out, you can take this back to your leadership and you can talk about how your room should be designed if there's deficiencies there. Because when there's deficiencies in room design, there's deficiencies in processing. So what should you have, or how should your room be? Well, it should definitely be separated physically from patient care areas and procedure rooms. For those that have been in the field for years, I'm sure you have seen processing done in patient care areas. You've seen processing done in procedure rooms. Well, that has been an issue. So your processing room should be physically separate from that. The room that you process in, it should be for processing only. It shouldn't be for storage. It shouldn't be for your lunch, and things like that. It shouldn't be your hangout spot. It should be for processing only. Just think of a decontam room in your standard sterile processing. What are you doing there? You decontam and you get out. You're not storing lunch. You don't have your sterile items there. So it should be designed just for processing. Designed for unidirectional flow of devices from the moment you receive the scope directly to storage to next patient. So there's a few options that you have, right? You have the 2 separate rooms for processing. Now, your area or workspace, you may not have the space to do that. But until that happens, there has to be a strict flow. You have to have it mapped out for some areas. In one of my areas a long time ago, before we went through our room design, I had to make sure that there were signs all over the place to say, hey, this is a soiled area, this is a clean area, and this is a high level disinfectant area. That way, if folks walked in, they knew what they had to do. They knew where they were supposed to be. That way there was no back and forth as far as the workflow is concerned. And you have to hold strictly to that. If you have a single room, some folks didn't know that you need a minimum of 4 feet of separation between the decontam sink and where your high level disinfection or your other area. So I've seen some areas they've had a wall. Some folks have had a plexiglass put up from their engineering. So this is to avoid splashing or recontaminating things that you have that you consider to be clean. You need adequate space for cleaning and rinsing. If you are in a single room, it's very difficult, but do everything that you can to make sure that you have that spacing. Now, this is something that folks may find new, and that's the minimum 2 sinks for manual cleaning and for rinsing. Again, here we go. You may not have adequate space to have these 2 sinks, but again, this is what you should have. So if you are able to do so in your facility, it would be wise to do so. Just think of the standard decontamination, right? What do you have? You have the 3 sink process. You have, and it's designed that way for a reason so that everything is not taking place in 1 sink. And we're talking about flexible scopes, difficult to clean. And so you want to have the separate environment for each step. But again, this is how it should be. So see how your area is and see what you could do to bring it to this. Next slide. Back to the design of the processing area. So countersinks and work surfaces should be height adjustable. I know for older builds and things like that, there's those standard sinks. I'm 6'4". I used to have a problem out of those standard sinks. I have an employee now that's 6'6", and we are just now getting height adjustable sinks. So this is great for staffing that vary in height. And it's all for ergonomics, right? Because we want to make sure that when you're in there processing, you're as comfortable as you can be. You want to make sure that the sinks and the countertops are sufficient to accommodate the scope length. You have some scopes that are small, and then you have those that stand taller than 6 feet. So you definitely need the space when you are processing or your workspace or your work area. Anti-fatigue mats. I've gone to some places where they haven't had them. And when you put them into your processing area, when folks are standing at the sinks or they're standing at the AERs, for a long period of time, those anti-fatigue mats actually come in handy. There are a number of vendors out there that have those. So if you do not have them, look those up. Anti-fatigue mats, they will definitely help you as far as standing for a long duration of time. Pass-through window is recommended between decontam and clean processing areas. Again, think of your standard decontam room. There's those items that are hand washed that are pushed through the pass-through window. This is the same ideal process that you should have in your scope processing area. Why? Because if you have that unidirectional flow, you want to make sure that there's that barrier between the clean and the dirty.
Garland, we have a great question I think would be perfect here. What the difference is between a decontam and a clean processing room, and piggybacked on that, then how would you designate those two rooms as it applies to HVAC requirements? Maybe it's a decontam or scope processing. What would you call them? So maybe just tell people what the difference are between the two areas and how they might be designated.
Okay. So when you think about if you think about a decontamination room, that is where all of the soiled items are coming, right? They come in, they're soiled. That room generally, it should have, or as far as HVAC is concerned, that should be a negative room. So the positive air, it shouldn't be pushing out positive air through that room, because the idea is you want to take all of that soil out. It's similar to a bathroom vent, right, that you have, that some folks have in their bathrooms. And so your clean processing area, where your AERs may be, that area or your storage area should be positive air. So you don't want to mix the two. And there's some air balancing that's done in certain facilities. Some newer facilities, they're monitored by engineering departments. Some facilities monitor their airflow and air balance. But basically the difference is kind of like if you think about washing dishes or something like that. You have your clean side and you have your dirty side. I don't know if that was explained well enough to the person, Stacy, in the chat.
So Garland, you mentioned that the AER should be located in the clean processing area or room, which I agree with, and that's what we have in the standard. So the question is, does that mean rooms with AERs can have clean storage in them too? What are your thoughts on that?
So there's the question about the clean scope storage. There's the thought when we talk about, and we'll talk about that later as far as scopes being dry, right, before they're stored. I've seen some areas that have a scope cabinet in their AER area for the sake of drying before they go to the end user department. Sorry about that. So as far as the clean scope storage in the AER area, again, this is a flow of who's coming in and who's going out of that particular room. If you have end users coming in that room to collect scopes, it would not be wise, because where are they coming from? If you have a workflow where it's just you and your department that is in that room, then that's a completely different story. But again,
right, Garland, and I like to think about it as best practice would be no, you wouldn't have storage even in the AER room, even though it's considered your clean processing area, because those scopes are dirty coming into there. They've only been cleaned. That doesn't mean they've had a terminal process on them. So best practice would be to not have it in there. And we did have two questions in the chat and the Q&A in that. So I wanted to make sure we cover that. In the meantime, I don't think we specifically say they can't be in there in st91, but while Garland's talking, I'm going to peruse the document and see if we do say anything else about not having clean storage where your AER is. So go ahead. I'm sorry. Right.
Go ahead. Next slide. So we talked about traffic control, right? As it pertains to the clean scope storage in the AER area. And this should be with all of our entire department, whether it's the decontam side or the clean side. It should be restricted to traffic as far as traffic access. And that includes engineering. That includes folks that are coming to repair our equipment. A lot of times, if we're not careful, they'll come in. Even the folks that bring us our supplies, they have to make sure that they adhere to how we have our traffic flow set up. Space requirements, sufficient space for each processing function, be that clean, be that ATP testing if that's something that you're doing, be that storage, sinks, dedicated plumbing and drains. And this is, and this here, electrical systems, ceilings, doors, HVACs, lighting, all of this is something that this document doesn't just pertain to the processors. This also pertains to the facility as a whole. They actually look at these. They see where the guidance is as far as how the HVAC system should be set up, the electrical system should be set up. Because as processors, as management, we don't go and determine and fix electrical systems. So this document applies to them as well. So they actually look at this as well when they say, hey, well, what document do you guys follow? Oh, well, we follow aami. Okay, well, what does aami say about the electrical system? What does aami say about the ceilings? They follow this as well. Next. Hand hygiene and eye wash and shower. Now, regarding the facilities, the sink, soap, towels, alcohol-based hand rubs, they should definitely be available. If you do not have adequate sink or soap or even the alcohol gel or reach out to your folks, get this in your area. The sinks that you decontam in the scopes in should not be your hand washing sink. If you do not have hands-free, see if your facility can get you hands-free, because you don't want to recontaminate your hands after you've washed them by touching the handles. Eye wash stations, this is a must. Anywhere you have chemicals, you need an eye wash station. They should be tested weekly. They should be less than 10 seconds of travel time from the location of where the chemicals are. It should not be in a separate room. You shouldn't have to walk out of one area to go into another area to use your eye wash. Your eye wash should not be in a decontam sink as well. And the temperature of the water, again, this may be an engineering function in your facility, because they have to test and they have to make sure that it meets these requirements for a comfortable flow and a comfortable temperature. We do not want to have to use the eye wash and the water's too hot or too cold for us. Next slide. Environmental cleaning, multidisciplinary team. What does that look like? Multidisciplinary team as far as environmental cleaning. Well, they define the roles and responsibilities for how your area should be cleaned. The agents that's used for cleaning, the equipment that's used, the frequency for high touch areas, terminal cleaning. How often should that be done? Define where food and beverages are permitted. We spoke earlier. I've gone into some areas in their processing areas, and there's food and water and drinks in their area. Well, should they be there? Again, you need that multidisciplinary team to come together that can include infection prevention, risk management, and quality as far as how these rooms should be cleaned and where items should be stored. Contaminated work surfaces shall be decontaminated after completion of procedures or after any spill of infectious blood. So what do we see here? We see that word shall. So there's no getting around that. This has to be done. What about staff, personnel? Some time ago, years ago, I know there was folks processing scopes, folks doing high-level disinfection that had no formal training, no competency verification. They were trained by someone a few years ago, despite changes in scopes. But this document clearly states that personnel performing processing shall complete formal training and competency verification in all aspects of processing prior to independent completing the task. Should be certified in scope processing within 2 years of employment and maintain that certification. Personnel should be provided education, training, competency verification on new scopes that come in, new equipment, new chemicals, and products. And this is important because one size does not fit all as it pertains to scopes. We just had a change in my facility with a scope. And it seems as though the cleaning would have been simpler. However, it wasn't. There was another 26 steps added to it. So we can't go with the idea of it's a new scope, we don't need training on it. No, we do need training on it. Whenever any new product or any equipment comes in, we should have training on it. And this is something that should be done. Address the responsibilities of those providing training. I've had surveyors come to my employees and ask them, well, who performs your training? And then they say, well, he performed my training. And then they'll ask me, who performed your training? Where did you get your information from? Years ago, they wasn't asking that question, but now they are. Because it's important, if you are training someone, you have to first know what you're doing before you try to train someone. Next. Back to hand hygiene. Some folks don't think that something as simple as hand hygiene should be part of a policy, but it definitely should be developed with your infection preventionists. Folks have fingernails. Some folks have very long fingernails. But that the policy should address how short your fingernails should be, not to extend beyond the fingertip. No artificial nails. I know some folks like artificial nails. A policy about nail polish, that should be in there. What should that look like? Hand washing per CDC or WHO guidelines. What is that hand washing? How does that look? Is it just rubbing this part of your hands together and calling it a day and rinsing it off? No. What does CDC say? What does WHO say? Hand hygiene with soap and water when hands are visibly soiled. What about alcohol-based hand sanitizers? How often should they be used before you have to physically wash your hands? Hand hygiene before you glove and after you glove. All of that should be included in the policy. Surveyors are looking at policies and they want to know not only that you know what you're doing, but they want to make sure that you are following your own policy for your facility. Next. Attire. Personnel should change into clean uniforms provided by and donned at the facility. We see all too often folks are wearing their uniforms home, bringing them into the facility. This document and other documents call out that you should change at the facility, change it daily or when soiled. They should be laundered by the facility. I know there's been some conversation about how they're laundered, but this should be done. Jewelry, watches, earrings, and things like that should not be worn. Shoes. You want to make sure that you have comfortable shoes, no open-toe shoes. Wear cover apparel when leaving the area to travel into another area, and remove the uniform before leaving the facility. Because what you don't want to do is you don't want to bring what you decontaminated home to your family. It's just as simple as that. PPE, you must have a OSHA bloodborne pathogen exposure control plan, which includes PPE. Now, personnel should wear PPE regular hospital laundered PPE, and also the additional PPE. Utility gloves, liquid resistant covering with long sleeves to the wrist. As far as processing in the in a decontam sink, you definitely don't want to use exam gloves. You want the extended cuff gloves. There are a ton of those out there. You want them to extend far above the wrist so that when you are processing in those sinks that are deep enough to process scopes, you don't get any of that water on your hands. So the highest level of gown should be worn. There's a chart for protective barriers that we'll consider in this document.
All right, thank you, Garland. I'm going to take over for the next couple slides. We're going to highlight the decontamination process next, which will lead us into the actual cycle of processing because we didn't even get there yet. But the document does highlight that your cleaning process, as we've all known, is the most important step in the process, because if we don't get that cleaning done properly and adequately, then we don't know if our next steps of disinfection or sterilization will be effective. And there is data out there in literature to show that soil remaining on scopes after the cleaning will lessen the effectiveness or completely inactivate the sterilization or disinfection. So we need to follow all of the steps of processing, follow the IFUs for the endoscope, adding in these best practices and quality parameters that we have outlined in st91 for all steps along the process. We do clearly point out at this point in the document that if you have endoscopes that are so old that they cannot be submerged for a leak test, they were never engineered that way, then they should absolutely not be used, because that is an infection control risk, because we can't tell whether those scopes are actually resistant to the water and fluid invasion or not. So definitely don't want to use those scopes anymore. And unfortunately, I do see that once in a while when I get into audit facilities that they're still using scopes that are that old, that have been discontinued for many years and are not supported by the manufacturer or other repair companies. One thing to hit on before we actually get into the process of the cycle for endoscopes is water quality. We have a lot more guidance in st91 surrounding the water quality that should be used for processing endoscopes. We have a recommendation captured by a should that facilities should monitor and control that water coming into endo processing sinks and to any of your endoscope processing equipment. And that would be doing tests for pH, hardness, alkalinity, just your general water testing, and that should be available in your department where scopes are being processed. This is a new recommendation related to water quality for AERs. We do recommend periodic microbial assessment. So that means bacterial cultures of the water, of your final rinse water within an AER, because there is evidence out there showing that contamination or biofilm within AERs can happen. It does sometimes happen, and that will then, we'll say, artificially contaminate, but will contaminate your endoscopes. And they've already gone through the high-level disinfection process, so there's no other step after that besides drying them that would then kill those organisms. So we do want to check periodically how to do that, or what that water quality is. And this has been done elsewhere in the world for a long time, where they will monthly do a test on their final rinse water to demonstrate that they don't have a biofilm or contamination issue within that. So we don't have a defined timeline or frequency for that, but we do say periodically that we would recommend that you test that final rinse water. Okay. So this slide is a little bit out of order, so I'm not sure what happened there, but we'll start with the first step in the process, which was formerly known as pre-cleaning. So now we've started in the cycle of scope processing. We've done the patient procedure. Our first step in the process is point of use treatment. So we've renamed that step to align better with st91 and other standards documents that call it point of use treatment. We really wanted to get away from the connotation that we do full cleaning at this point in the process. We don't, right? We do a wipe down, we do a suctioning, flush, but we are not doing full cleaning with a cleaning brush, and we don't ever want to do that in the procedure room. So this is preparing the scope for further steps of processing in the endoscope processing area. So that's new with the terminology. What else is new is that we have a recommendation in there to note that time that pre-cleaning is performed. So when you do your point of use treatment, what time is that? So somewhere you need to note that and then convey that information to your processing staff. We call that handoff communication. And the reason that we have that in there, just like the scope manufacturers have guidance on that, we have aligned with that recommendation to do this documentation of time. We're not looking for official log or an official document like that. We're looking for somehow it might be a tag on the endoscope or a label on the transport container or in your electronic record, however it may be. But your staff that's going to further treat that scope by manual cleaning and all of those subsequent steps needs to know, are they within a 1 hour hold time from the pre-cleaning to the manual cleaning? Because if we are over 1 hour, then we do have to follow the delayed reprocessing procedure. That is in the instructions for reprocessing of the endoscope from your major scope manufacturers. And it's not optional. If that scope sat for longer than 1 hour, then you have to follow that process as outlined in the manual. The problem with that is a long-term submersion in the detergent solution prior to even starting manual cleaning. And you do have to look at your type of scope and the manufacturer to know what exactly the guidance is. But generally, it's soaking for up to 10 hours for your GI scopes and up to 1 hour for your surgical or small diameter flexible scopes. That's a long time, and you're going to tie up your sink. So we want to avoid this as much as possible, that longer than an hour delay. So the noting that time and conveying will help stage scopes appropriately, knowing which ones have to get done when, because we don't want to do this delayed reprocessing, because long-term submersions on a frequent basis can lead to damage of the endoscope. So let's avoid that as much as possible by actually having an objective measure of that time between point of use treatment and manual cleaning in the scope room. So then we're going to go to transport of soiled endoscopes. So this is captured as a must within st91. So that means a requirement. In this circumstance, it's by OSHA that we should be containerizing either in a solid container or a transport cart, and that container itself must be labeled as biohazardous, and we have the normal requirements: an orange or orange-red label, fluorescent label, the marked biohazard, and it should be in that closed container that's non-porous, has leak-proof sides and bottoms, and is puncture resistant, and also large enough that the scope can lay in its natural coils. We don't want it in a container that's so small that we're causing damage to the endoscope. That should be isolated from other components, never multiple ones in the same container. There is a recommendation in there that the endoscope be kept moist but not submerged. And this was actually in the last st91 also, but not a lot of people embraced this recommendation, but we did keep it in there, moist but not submerged for transport. And that is to keep those soils loose and hydrated so that they're more likely to be removed adequately during the full manual cleaning process. And again, we don't tell you how you have to do something, just what should be done. And then it's up to your facility to put a policy in place around that and pick a way to do these things. So we give examples like a pre-treatment solution or a water, not saline, moistened towel, or a humidity chamber bag for transport. And that does not allow you to get away with doing the delayed reprocessing unless you would have that evidence and validation from the manufacturer. But at least it keeps those soils moist for transport that we can do a good job then during cleaning. All right, next step in the process is leak testing. A lot of new information here, critical that it's implemented. Of course, I said before, you should not use endoscopes that cannot be leak tested. I'm not going to spend any more time on that. We have changed the time of observation for leak testing from 30 to 60 seconds. And that's because it really takes a full 60 seconds to do a high quality leak test where you're massaging all the buttons and you're manipulating the levers and turning all the knobs. And per the IFU, you're running your hands over the scope. It really does take that long to do a critical evaluation to see if there are any bubbles anywhere coming out of that endoscope. So that's a big change, and that more aligns with what the rest of the world is already doing for a leak test when you do a wet leak test submersion. What else is new is related to the leak test units themselves. So we have there are many different kinds on the market. The most common ones are still the little box that sits on your counter and it pressurizes the endoscope and you submerge it in under the level of the water only, never detergent at this step. And I still see that done, unfortunately, too commonly. So that's most commonly what's used. But those leak testers, the pumps can wear out on them. And right now, the only quality indicator we have, it says in the IFU, is to push the pin and listen for a psht sound. But that doesn't really objectively tell you whether it's working properly or putting out the right amount of pressure. Because as I said, the pumps can wear out over time. There are O-rings in the connectors. Those are considered consumable items as far as the connector, the black cord that you see in the photo there, and they should be replaced when they have signs of damage or wear and tear. So we do now say that you should inspect your leak test units for damage and leakage and the correct pressure output. So what that means is, as far as the standard goes, we now recommend daily, every day that it's used, that you verify the output pressure from that leak tester and document that. So that is something we're asking to be documented. I've seen many, many instances where I go in and test leak tester, and sometimes they're making a sound and they're not outputting any pressure at all because an O-ring went somewhere and all that air is escaping, or something else is damaged. So there are various different testers on the market. You can work with facilities or your biomed to figure out how you're going to do it, but test your leak testers and document it and make sure that they're regularly calibrated. As far as manual cleaning goes, we didn't put much more new in here because we really focused on that in the first st91. We stated it's critical. It needs to be done immediately without time delay. We've discussed the delayed reprocessing procedure already, but as scopes sit and they're dirty still, right? They're still highly contaminated. You can have these biofilms start to develop, and then we don't know whether we can ever get those scopes truly cleaned, decontaminated, disinfected, or sterilized. So we have to make sure that we're avoiding this biofilm formation because it is very tightly adhered to the surface and extremely difficult to remove once it's there. And that's why we need to make sure the subsequent steps of endoscope processing are followed in order and without time delay as often as possible so that we don't have these biofilms start to develop. The manual cleaning process, we recommend that you follow the IFU for the endoscope model that's being used, making sure we're using proper cleaning solutions. Those are our detergents, proper accessories. I still see facilities that aren't using things like the air-water channel cleaning adapter, or they're not doing suction and they should be doing suction. You're following the IFUs for any detergents that are used, any additional processing equipment like your automated flushing pumps that flush water, detergent, water, and air through those endoscopes. You want to follow all of the quality parameters for that and testing that. But manual cleaning is a really thorough set of steps, of sometimes over 100 steps just for manual cleaning. It involves wiping the outside surfaces, brushing all the channels that can be brushed, suctioning, flushing, all of that to remove that residual debris from the scope so we're more likely to achieve disinfection or sterilization. And even though there's not a lot of safety margin built into disinfection, that's why we need to make sure that that these devices are being cleaned adequately every time. I see a question. I can't look at most of them, but how do you verify a dry leak tester? There are some products that are on the market that can verify even dry leak testers. So you'll have to do your homework and look at what those are. As I said, lots of steps within manual cleaning, and we didn't change any of these per se, just went through and made sure they were adequate, follow the IFUs, making sure that you're doing everything appropriately, and following your instructions for your leak test also per the IFU, just upping that time to 60 seconds instead of 30 seconds. Make sure your detergents are compatible, that they are made to process endoscopes, and that you have that compatibility information from the detergent manufacturer. Always use fresh cleaning solution. We don't ever want to be reusing detergents in our sinks for endoscopes. It's for, think about it as single use for really everything, whether we're even for reusable brushes and things like that, we still need to reprocess them in between use. So one scope, one basin of detergent, one brush, all of that. No cross-contamination or cross-use of these items between different scopes. Follow your detergent manufacturer's instructions for what temperature it should be used at. You need to monitor that. Make sure that it is appropriately diluted per the IFU or per that label of the detergent, and what quality of water you should be using. Make sure that it's completely immersed under the level of your detergent and water solution in the sink. I know that sounds intuitive, but I was just at a facility not too long ago where there was not nearly enough water to fully cover that endoscope for cleaning. And make sure we're wiping down the outside surfaces of that scope with a fresh, so new every time, single use, non-linting cloth or sponge to do the external wiping parts of manual cleaning. As far as cleaning brushes go, we do say, and in line with what FDA and other professional societies say, is that you want to make sure you're using a brush that meets the guidelines or the guidance from the endoscope manufacturer. I'm not saying you have to use that exact model of brush, but if you don't use that model, then you want to make sure it matches those characteristics of that brush as far as length or width of the bristles and all of that. We don't require that they have to be single-use brushes, although I will tell you that the majority of facilities are using single-use brushes. But if you're not, then the brushes themselves must be cleaned and disinfected or sterilized in between uses. So that's important to make sure that we're decontaminating and reprocessing those appropriately, making sure we're doing everything right with that forceps elevator on our duodenoscopes, of following the IFU from our scope manufacturers for that, and the manipulation and flushing of it and everything, brushing at all steps along the process. That's very important to make sure that we're getting those types of scopes clean. Okay, I see a question for clarification. Wet leak testing is 60 seconds. Yes, that is true. And as far as dry leak testing goes, follow your instructions from your dry leak tester manufacturer to know what to do for those. The 60 seconds is definitely for the wet leak test. If you can get that instruction from the IFU for the dry, that would be the best case. Garland, over to you.
Alrighty. We have quite a few questions in there. Maybe we can, you took some, I believe.
Yeah, I think that Garland, at this point, we should see what we get to at the end. We're already over an hour into the program with lots of slides to go. So why don't we get through this? And I promise if we don't get to all of them today, Garland and I will divvy them up and get back to you with the answers. And for some of them, we are very familiar with the standard, but I can't always say I remember exactly what the guidance is for every topic. So some of them I may have to look up to confirm that I'm not telling you something incorrectly. Okay, go ahead.
All right. Now moving on to automated cleaning. One of the questions was about automated cleaning too. Automated cleaning manufacturers' written IFU should be followed. I know some folks have seen some where they say, just do this, but don't do that. There's a slide in here that talks about when one piece of equipment disagrees with the other. We'll discuss that later. Fresh cleaning solution should be used to verify the solution flows through each lumen as far as automated cleaning is concerned. Model-specific cleaning adapters compatible for that particular scope that you are using. It's not okay to modify the connectors. You want to make sure that you use the connectors that are meant for that particular scope that you're processing. All connections should be correctly secured to the endoscope, so you want to make sure that nothing pops off during the flushing cycle or the purging cycle. Channels should be flushed with water for the length of time specified by the manufacturer's written IFU. Now, what we do in this 91 document, we call out a lot. Some of you may hear a lot, best practice, best practice. Here we call out manufacturer's written IFU. You have to know what the manufacturer says for everything that you have so that you can come up with a best practice should there be some conflict. Next. Connection tubings or adapters should be cleaned and disinfected according to the manufacturer's IFU. So some tubing connect adapters, some say daily, some say weekly, some say after every use. Whatever the manufacturer calls out, make sure you do it. Again, these surveyors are looking at these manufacturer's instructions. They know what we have in our facility. So when they come, they are looking. I just had a survey last month, and surveyor wanted to see the cabinet manufacturer's instructions, the AER manufacturer's instructions, and our sink manufacturer's instructions to make sure that we were doing what we were supposed to do. Suctioning during the manual cleaning still might be necessary. Again, follow the endoscope and pump manufacturer's written IFU, because you may see some scope manufacturers say the scope needs to be suctioned while your sink or automated cleaning area may do flushing. So follow those and combine for best practice. Next. Back to identifying these high-risk scopes that we mentioned in the outset. The ones that we talked about are associated with infectious outbreaks. They're difficult to process. Some of the older model scopes in their design, they had no exit as far as brushing. Some had lumens where there was never even a brush designed to go down the lumen. And so ideally, you had to flush those lumens as best as you can manually. But again, some of those scopes that have been identified as high risk are duodenoscopes, EUS scopes, bronchoscopes, EBIS bronchoscopes, ultrasound bronchoscopes, ureteroscopes, and cystoscopes. So it's good to check your facility again, see what you have, see what your risk is, see who's processing. You know, there's so many different processing areas. You may have dedicated processors in one area, and then you may have 50 people in a urology area that are processing. So identify your risk in your facility so that you can make sure that you're properly taking care of these scopes. Next. Cleaning verification testing. Now, you definitely want to make sure that you are doing cleaning verification, in particular on those high-risk scopes. Again, you say shall be monitored with cleaning verification after each cleaning and prior to high-level disinfection or sterilization. Scopes not high-risk scopes should be verified using cleaning verification when new endoscopes are purchased, and however you deem your percentage rate. So you may want to do 10% of your scopes. You may want to do 5%. You may want to randomly pull scopes from sterilization. You may want to randomly pull them from your scope cabinet. But you want to just do this cleaning verification so that you can ensure that the process that you have in place is actually working. And if not, this is where you make the needed changes. This is where you make the adjustments. There's an annex in 91, Annex F, that talks about frequency and determination, considering the factors, the type of scopes, the type of procedures. And again, we go back to the technician and their competency level. You have some that have processed for years, and you have some that have processed for months. So And then also delayed processing. It's good to do cleaning verification on delayed processing. How good is your process when you get a scope that has not been cleaned within that 1 hour, or the process has not started within that 1 hour?
Garland, we have a ton of questions coming in asking about clarification to this and what exactly we're talking about for cleaning verification. So I thought it worth taking a step back and talking about what type of testing we mean here. Not knowing the audience and what their backgrounds are. Maybe we can take a minute. I don't know if you want me to take this or if you want to. So a cleaning verification test or a cleaning monitor is a type of test that's done after the manual cleaning process, but prior to your putting it into an automated endoscope reprocessing machine, an AER, or prior to sterilization. So it's always meant to be done at this point in the process after manual cleaning, because you're verifying that your soil is either not detectable or has been lowered to a level that can't be detected at that test. So you're more likely, if it passes a cleaning verification test, to go on and actually achieve high-level disinfection or sterilization. So we had it as a recommendation in the last st91 to periodically monitor, and then we meant weekly, preferably daily is what we said. So we've really amped that up now. For the high-risk scopes being a shall, as Garland said. So that is something that you would have to do per the standard every time for every high-risk scope. And then for non-high risk, as he said, should verify those on a pre-established interval. And we have an annex there to help you determine what that frequency is. And as far as other questions, like, what types of tests are these? People don't even understand what that means. So these are tests, a wide variety on the market. So you have to look at what works best for your facility, for what marker it's looking for, what the detection levels are, how easy is it to use, how reliable, what the price is. All of that you should consider when looking at the type of test for cleaning verification. Examples of these are tests for residual protein, tests for residual hemoglobin, blood. There are combination test strips that look for protein, carbohydrate, and hemoglobin, and ATP. And that's a question I have here. Is ATP an option? Yes, it is an option, one of the many, and this would be where it's used after manual cleaning. So I think that takes care of most of the cleaning verification. Okay, asking for examples, we did that. Ways to monitor, we talked about that. So following the instructions from whatever cleaning verification test you're using, but making sure we're doing at this point in the process to show that you've achieved that cleaning level. Sometimes we see them used after disinfection, and that's not appropriate. No cleaning verification test is meant to be done at that point. It's on a scope that's been cleaned, but not yet terminally sterilized or disinfected. So sorry for that interruption, but I was realizing that people didn't know what we were talking about.
Thank you.
Thank you.
Next. Visual inspection. Again, endoscope should be inspected with each reprocessing cycle. Lighted magnification should be used for inspection for debris and damage. FDA recommends 5 to 10 times magnification for duodenoscopes. Some of our areas, the lighting is poor, and it's due to old build and things like that. Even in my area, we have been upgrading and doing more to get this lighted magnification, and it's done a world for staff. Some of these scopes are very small. Some of them are large enough where you can see, but with our imperfect eyes, even with glasses at times, we still can't see. So if you do not have it, get lighted magnification in your area. Next. Borescope inspection. Some folks have had a borescope. Some folks have heard about borescopes. But borescopes, we discussed this as far as its purpose and checking the internal channels. And you can use the borescope. There's many different sizes out there that you can use for the different scopes that you have. You can use it periodically. You can definitely use it on your high-risk scopes. You want to make sure that the scope is dry prior to inspecting, because what happens is when you drive that borescope down that lumen, you will see all the water droplets that's there. It's actually a good way to test to see how your drying is in your facility. Are your scopes dry when they're hung? Are they dry after 4 days of being in a scope cabinet? If you have a scope that you did cleaning verification on and it continues to fail, it will be wise to use a borescope to see what is really going on inside of there, because there's been times where we've had to put a borescope down a scope, and we see that there's small tears inside of the lumen, and that could have been due to instruments being placed down the scope during use. But this is an option that you have as far as inspecting the internal channels of your scopes. Next. Spalding classification. This is just a quick chart on the Spalding classification. This has been a topic of discussion over the past few years, and what that looks like, and what does that mean for non-critical, semi-critical, and critical. How should they be handled? You have blood pressure cuffs there, which patient contact intact skin, which is considered non-critical, mucous membrane or non-intact skin. You see scopes, specs, semi-critical, cleaning and sterilization. Now, if sterilization is not possible, then definitely high-level disinfection at minimum. Regarding sterile areas of the body, definitely critical, cleaning and sterilization, period. Next.
Garland, one important thing related to, or a couple that I see here, related to borescope usage, a question of where it would be used. We don't stipulate in the document exactly where it has to be used. So I will tell you some facilities use it as part of their inspection process after manual cleaning. Note that you have to dry the scope then before you'd be able to see anything besides water in it. And some facilities will use it as a quality monitoring tool, where they occasionally with some established periodic interval, will take a scope out of the storage cabinet and inspect it to show that they're in a good repair condition, and that their drying is working properly. So they could be used at either place, depends on your internal processes. So that takes care of a couple questions that I saw. And then is borescope a must, shall, or a should? It's actually a can, so we have it. It's possible for you to use. This was a big area of debate, and many people felt we should have it as a should or even a requirement, but it's a consensus standard, so we landed on a can, and that we do recommend that it be used, but it's not required. Just make sure when you're using a borescope that you're reprocessing that borescope per the manufacturer's instructions for that device. And also think about if you are inspecting scopes that have been through the process that you've taken out of the cabinet, that we do recommend that those be processed again before use. So you're really doing a quality check of the process itself. Okay, I thought that was important to interject there.
I was going to mention that. Thank you. So this is basically the traditional processing for scopes. Once it's used, you go through your pre-cleaning, or what is now point of use, leak testing, manual cleaning, rinsing after manual cleaning, cleaning verification, inspection, high-level disinfection, drying, and storage. So this is just kind of like the life cycle or use cycle of a typical day in the life of a scope.
Garland, just one more thing here. A question about what does damage look like, and most staff don't know what to look for. We do have a great pictorial annex within st91 that'll help walk you through points to inspect and what to look for, with lots of photos in there. There are great training sessions available as free webinars online. The Ofsted group comes to mind as having webinars that address internal inspection of endoscopes as another thought on that, and really look at the training that your borescope manufacturer can give you.
Thank you. I was going to address that too, because those pictures are absolutely amazing. High-level disinfection. All flexible scopes that are introduced directly into the bloodstream shall be sterilized. Now, so again, think about the scopes that you're using. Where are they used in the body? What process should you follow? Again, consider those high-risk scopes also. HLD is minimum standard of care for semi-critical devices, even though sterilization should be the goal. Now, that is our stance. In fact, everybody feels the same way, that sterilization should be the goal. Everybody's not there. Again, we talk about room design, spacing, and in some cases, financial. It's the financial aspect of moving from high-level disinfection to sterilization. So, but ultimately, sterilization is the goal. So semi-critical devices, again, going back and speaking more on that chart, contact mucous membranes or non-intact skin. Definitely thorough cleaning and then sterilization. If sterilization is not possible, here we go again. Minimum high-level disinfection. Okay. We did not require, the 84 group didn't require sterilization for all scopes because all scopes can't be sterilized currently. Certain manufacturers are catching up with the view that many have as far as sterilization of flexible endoscopes. We advise that all flexible and semi-rigid endoscope be sterilized prior to use. Transition from HLD to this process. It's no different than instruments. When you're having a procedure, you want sterile instruments. The reality of the matter is you want the best care that you possibly can, and sterilization is the direction.
Totally agree with that, Garland. And it was a lively discussion amongst the group on this topic. And, we did stop short of requiring sterilization, as he said, but we do really want to be striving toward that and trying to maneuver or push the industry to having devices that have better compatibilities and clearances for new types of sterilizers and all of that, I think, is going to help move us to that over the next couple years. Moving on to another a big topic that I think is going to have a huge impact on our group here is manual disinfection. So we no longer recommend manual disinfection be performed at all. We do have that in there in like an emergency situation, if your AERs went down and you still have procedures that have to be done. But generally, we want to move the industry to reprocessing scopes in automated systems in your AERs, because there is so much evidence out there to support that your AERs are going to do a better job, a repeatable process. They're going to flush higher volumes of fluid and like your disinfectants and detergents and all of that through the scope than we could ever do manually with a syringe. So we do want to think about those implications. And as I said at the beginning of the program, look at where your scopes are being reprocessed. This is going to really affect your off-site locations where they're doing manual soaking in the disinfectant. So it might be that they're soaking in OPA still, or they're soaking in a glutaraldehyde in a container with a lid, hopefully. So that's no longer recommended for routine processing. So it's going to be a challenge. I know some people have space issues, and we know that it's a financial issue too, but this is something that we definitely want to get facilities moving to, is this automated process. We're alleviating that variability and inconsistency that is going to happen anytime humans are involved. And like I said, just so much evidence out there to support moving to an automated system. Drying is another important topic that we've put a lot more information around, and I know we're getting a ton of questions that we're not going to have time for today. But Garland, maybe an idea is we take all these questions and we highlight those as one of our next training sessions, or part 2, or just going over questions.
That is exactly what I just wrote down.
Yeah, because remember, this is not an addendum to st91. This is an entirely new document. It's a replacement for the 2015 version. So it's going to take time to digest all of the changes that are in there. So I think hitting on these refresher courses is going to be important to get really into the weeds, because we couldn't cover everything today. And I feel so bad. You have so many great questions. But drying is such an important topic. We want to make sure that these scopes are completely dry prior to going into a storage cabinet or prior to being reused. And this is something, again, that we had a lot of discussion around the topic of drying and when is it appropriate to completely dry and when is it not. But what we landed on is all scopes. Every scope, every time must be completely dried with compressed air. We do recommend instrument quality air, and that's defined within the standard, at minimum HEPA filtered air for flushing. And generally we recommend 10 minutes if you're doing a process with like a drying pump. We don't recommend that it's not just flushing with a syringe anymore. You have to do this pressure regulated forced instrument quality or minimum HEPA filtered air, as I said, on all scopes. So yes, that includes those coming out of an AER. They don't have drying cycles. They have air purge cycles. Those scopes are still wet. Yes, even if you've done an alcohol flush on them. Yes, even if it's going into a regular storage cabinet. Or it can go into a drying cabinet. And a drying cabinet are those hooked up with active air lines directly to the channels that either continually blow air through those channels to dry or have a validated cycle to achieve drying. So again, every scope, every time, completely drying it. It doesn't matter. And I know we'll get probably 10 questions on this. Oh, my scope's going to be used immediately. I'm taking it right back into the next procedure room. Even those have to be completely dried. So think about that time. We already talked about additional time for inspection, additional time for cleaning verification, now additional time for drying every scope. You automatically are going to have lengthier cycles. And I can't tell you exactly how long it's going to be. This step might be 10 minutes, or if you have a drying cabinet, it might be an hour that they have to be in there. You're going to have the additional time for doing your cleaning verification test and waiting the appropriate amount of time to get the result on that. Time to do your inspection. So we need to think about scheduling our procedures appropriately and making sure we have enough inventory of scope. This is going to have big impacts, and drying is included within that. So again, just to reiterate, because I do see questions on this too, every scope must be completely, adequately, thoroughly, whatever you want to say, dried every time. Again, doesn't matter if it's been done in the AER, or it doesn't matter whether it's going back for reuse immediately. Every time. We have to dry externally too. So the standard says using a clean, single use, either clean or sterile, because some of the scope manufacturers, IFUs, do say sterile. We stop short of requiring sterile, but we say clean or sterile, single use, non-linting cloth to dry externally, and whatever else your endoscope manufacturer says. Sometimes they tell you have to dry out the valve housings or around the forceps elevator. So of course you would want to do all of that too. And think about your alcohol flush. There are some guidelines out there that no longer recommend doing an alcohol flush. We know that some of the AERs have that automatically as part of the system, but what we recommend is a multidisciplinary team evaluate whether or not you still need to do that alcohol flush. Because if you're doing everything per the standards, guidelines, and IFUs, adding in all these quality parameters for cleaning verification, inspection, now drying. So why would we still need to do the alcohol flush when we know it's not drying the endoscopes? We have a lot of evidence on that. I've inspected scopes that are hanging in a storage cabinet for a week, and they had an alcohol flush done on them, and they're still wet inside. Doesn't dry them at all. And alcohol is a fixative chemical, and we now have evidence supporting that. So why would we still recommend drying? Well, it's still in the IFUs, and it's still part of the cycle for some AERs. That those all haven't caught up yet. So we do recommend that multidisciplinary team look at this as a topic and decide whether that's something you still need to do as part of the drying cycle for endoscopes. Okay, storage, another important topic. As I said, we did stop short of requiring a drying cabinet because we know that's not feasible for everybody. And if you're using some kind of drying pump, there are many of those on the market, then that's adequate if it's getting your scopes dry. But we want to make sure that we're verifying that periodically too. So we do have a recommendation in there to verify on a periodic basis, whatever you deem that to be, that your scopes are actually being dried by whatever type of process you're using. And there are different drying indicator tests that are available on the market. So you'd set what that is that you're going to use and at what interval, and checking your different kinds of scopes, right? I'm not saying every scope every time for this, but it might be monthly or weekly or quarterly that you do a dryness test on a verification test on your different types of endoscopes to show your process is still in control. At minimum, if you don't have a drying cabinet, you should have HEPA filtered air circulating within the cabinet. So we shouldn't be using ones that don't have any ventilation or any HEPA filtered air going through it. We kept our storage recommendations the same as they were before, which is in line with AORN, which is that you should do a risk assessment on the topic. And we have a great chart in st91 that helps you walk through the topic and look at your practices to help identify what your storage interval or hang time should be for your endoscopes. Because if we're doing all these extra steps and quality parameters and we're completely drying that scope in there before it goes into the cabinet every time, then maybe we can get a longer storage interval, but that's something you need to evaluate internally. We kept the recommendation to have a distinct visual cue on your endoscopes so that anyone or I can walk into your room where your scopes are being stored and see visually that they've been processed. So that recommendation we still have in there as a should. Making sure our cabinets are located in a secure location, like a clean workroom, and not the procedure room. And I see that a lot in the ambulatory settings where the storage cabinets are right in the procedure room, that we should try to move if we can. And making sure we're cleaning the cabinets on a regular interval, at least weekly. Some facility, that's what I say most are doing is weekly. Some are doing it daily. But inspect your cabinets too. These were just two pictures I took at a facility. It was visibly dirty in the left cabinet, and there was some kind of residue on the floor of the right one. I don't know whether that's residue from a scope dripping alcohol out of it or from wiping it. I don't know, but it shouldn't look like that. Think about it, as a compliance issue when a surveyor comes in. They'd be looking at your cabinets for all of this too. Okay, next topic is, now that we've processed our scope fully, how do we handle that appropriately? So we did put recommendations in there for hand hygiene and glove usage. So really look at that section and digest it. We want endoscopes to be handled with gloved hands at all times. Of course, we would do that for contaminated scopes, but we should also be doing that for processed endoscopes because we're all notoriously bad at hand hygiene, and we don't want to artificially or recontaminate that device. So we say perform hand hygiene and don new, clean, non-latex gloves to handle processed endoscopes. So this is your clean transport. And where I often see the breakdown is not with processing staff. They're great about making sure they have gloves on to take them out of the scope out of the AER and dry it and put it into the cabinet. But often when staff comes in to pick that scope up and take it back to the room, that's where I sometimes see the breakdown. So making sure that all staff is aware of making sure we're handling these with clean gloves from the cabinet on back to the procedure room. They should also be containerized. Here is what we're recommending, that they're protected from contamination by putting it in a container, unless it's in a controlled connected corridor. So look at that wording within st91 if you do have questions. But if it's not in that controlled interior corridor, then you should be putting it in a container so that it doesn't get recontaminated by the environment. Making sure our containers are processed appropriately also. So you should have a container that has an IFU that tells you how to process it, how to clean it and wipe it down with a surface disinfectant wipe or run it through some sort of other terminal process depending on its compatibility. And we want to make sure that those containers help to prevent not only the environment from contaminating the scope, but also from damaging the endoscope. So it should be clean, covered, solid, like we say with contaminated transport, non-porous, leak-proof are what we're looking for. But really, let's treat these like the delicate, expensive instruments that they are and protect them from that recontamination. Okay. How are we doing on time? All right, we're doing great. So the next topic on the list is sterilization. And this is, in my opinion, some low-hanging fruit that you can take care of, even though not everybody's going to be happy about it or on board with it. But before we talk about sterilization, I wanted to show you these two workflows because they are a bit different when moving from high-level disinfection to sterilization. And if you look in the endoscope manufacturer's IFUs, normally at the beginning of that reprocessing manual, it has a workflow. So it tells you what steps you still have to do to get to high-level disinfection, and then what steps you have to do for sterilization. So normally those IFUs don't say you disinfect and then sterilize. You do the beginning part of the process. So you do your point of use treatment, leak testing, manual cleaning, rinsing. Then you're going to do your cleaning verification test and your external inspection with lighted magnification. You may be using a borescope at this point. You may not be, but then you stop there. Moving on to sterilization, then at the bottom, we see now we have to dry that endoscope completely before we package that in whatever kind of sterilization packaging that we're going to do. We move to sterilizing that. It might be a low temperature sterilization process, like vaporized hydrogen peroxide. It might be ETO if you still have that. It might be a newer type that has FDA clearance now. Sterilize that, and then you have storage. And what's great about moving to sterilization is you have a wrapped, storable item that can stay flat in its wrap per your healthcare facility policy on sterilized items. So whatever you have for everything else is what you can do with these sterilized endoscopes. And then we go back to reuse from there. So we see clearly by the IFUs, it does not high-level disinfection is not included as part of the sterilization process. You're really subjecting that scope to a lot of chemicals and will get damage potentially much earlier than you would by just doing one system or the other. Okay, so we know sterilization has a higher margin of safety built into it, but it is difficult, especially with certain types of scopes, to achieve sterilization. Example is your GI endoscopes, your regular gastroscopes and colonoscopes. There's not a lot of methods available for sterilizing them. But as a group, we felt, as Garland said earlier, that we do need to move to sterilization over the next few years because there is this higher risk associated with endoscopes and high-level disinfection. So although sterilization is not foolproof, it still requires complete and effective cleaning. We still have to completely dry that scope before sterilization. We still need to do our inspection and cleaning verification, but we just have that higher margin of safety. So we do recommend sterilization for all scopes, but as Garland said, we can't always get there. So what I recommend for sterilization is looking at the models of scopes that you have. So you should already have an inventory of what types of scopes you're using within your facility. But then I need you to dive deeper and take a look at what compatible methods are available or not available, are validated for that type of endoscope. And what I mean by that is, in the endoscope IFU and in your instructions or validation from your sterilizer manufacturer, they'll tell you what they've tested and what's validated for what systems. So look at your inventory and see what it's already compatible with. You may be surprised. So one of the first things I look at when I do audits of healthcare facilities is, what are you doing with your bronchoscopes? Most facilities don't think about it. They're putting them in the AER and going through high-level disinfection. Well, those types of endoscopes, the majority of them and vast majority of them have been compatible with vaporized hydrogen peroxide systems for years. For like 12 or more, 15 years, some of them have been compatible with those things like STERRAD and vPro certain cycles. So take a look at that. Like I said, I consider them the low-hanging fruit. If you have an endoscope that's already compatible with sterilization and you have that method available in your healthcare facility, albeit it might be in sterile processing instead of endo processing area, that scope really should be sterilized then. That is what the Spalding classification says, and it's already compatible, and you have it available, and you have a higher safety margin, and those scopes are going into a higher risk population that's already sick. So why aren't we sterilizing our bronchoscopes? I'm sorry, I'll get off my preachy wagon, but that's something I'm very passionate about. Is we need to have some of these scopes, we need to hold to a higher standard of care than what we've done in the past. So look at those high-risk scopes, your ureter scopes. The IFUs have already changed over to sterilization. Are you sterilizing that? Some facilities are not still, even though IFUs have removed disinfection. So take a critical evaluation of what you have and what you can move to sterilization as soon as possible. All right, culturing is another important topic. We did not require surveillance, microbial surveillance, which is sampling and testing of your endoscopes for bacterial contamination. I just want to make sure everybody is sure what I'm talking about here. I'm not talking about cleaning verification tests. So no, this is not ATP or protein tests or any of those. At this point in the process, we're talking about a full surveillance culture of your endoscope where you're following, the recommendation is the joint method from the FDA, CDC, and ASM together created a sampling and culturing method for duodenoscopes. Although that can be applied to other types of endoscopes, you would start with that method. And it tells you how to go through and sample the channels of that endoscope, flushing, brushing, flushing, getting that sample and sending it off to a lab. It might be a commercial lab, a third-party lab who can do that testing for you. There are mail-back culture kits. Your internal clinical lab may be able to take those samples and test them. Sometimes infection control gets involved in actually physically taking the samples. But although it's not a requirement, it is a good part of an overall quality control program that shows you that your endoscope process is under control, right? That's the main thing, that something hasn't gone out of whack and all of a sudden you have contamination in your endoscopes, because we do have evidence that was published from the FDA that there is always a certain portion of these. The last published report that we included in st91 showed a 5% culture positive rate for duodenoscopes with organisms of concerns. These are our gram-negative organisms like E. coli, Pseudomonas, Klebsiella, the remaining in those scopes after proper processing. So these samples were the healthcare facilities were watched when they were doing it, that they were following their process, and we still have this baseline level of contamination that it seems a challenge to get rid of. And I believe since then there have been more results published from the FDA. So we do have recommendations from the FDA to do more with these types of scopes, your duodenoscopes in particular. Sterilization is one thing. Culturing is another recommendation. Liquid chemical sterilization. It used to be 2 rounds of high-level disinfection, but you'll see in the latest recommendations from the FDA that is no longer mentioned. They do because there's data that shows it didn't matter. You don't get a statistically significant difference whether you run it one time through high-level disinfection in an AER or twice. So think about, what am I doing with my duodenoscopes? I really should be doing more with them per recommendations from the FDA. But on a routine basis, as far as surveillance goes, a good quality monitoring tool through this sampling method of full culturing of your duodenoscopes and then taking that sample and growing it out and identifying the bacteria that would grow in that culture. And then the FDA method tells you what to do depending on the number that are there, the type that are there. All of that is established within the FDA joint method. The other option for surveillance is a rapid gram-negative test, and there are some of these commercially available on the market. And then we did put the data in there from published studies, or the information from published studies that support the use of those rapid gram-negative tests as a screening tool. And by rapid, they're usually about a 12 to 24 hour overnight, potentially 12-hour turnaround time on these rapid tests, whereas your full culturing methods take a lot longer than that, depending on whether you're doing them internally or externally, and whether you have bacteria that actually grow on it, and identifying them. That can take up to a week or 10 days even to get those results back. So these rapid gram-negative tests are available and can be used on a more frequent basis, less costly, all of that. So we have that information within ST91. Okay, personnel training and certification. I did see some questions on this earlier, and we do have recommendations of certification bodies within ST91. We do say HSPA, which formerly known as ISHM, has an endoscope. CBSPD has an endoscope certification. SGNA also has a program available specifically for endoscopes. But we do have in here that all personnel shall complete formal training and competency verification on all aspects of endoscope processing, even prior to that first assignment. So before that, you should always have somebody working with anybody new till you complete that formal training and competency program in your facility. We also say that all personnel should be certified within 2 years of employment and maintain that certification throughout that employment. So we're asking that it be written into job descriptions even, so that it's established upfront that this is an expectation. And I know states are moving toward mandating certification for processing staff, but it runs the whole gamut as far as what state requirements are. So we put this in here as a should, but the competency and training, formal training program are a shall. Okay, sterile sheaths, a whole nother topic that we do discuss in detail in ST91. More information about the use of sheaths to cover endoscopes and what that does to the scope as a barrier then. So for sheaths that are used as a microbial barrier, we say that they are available and are FDA cleared for some specific types of endoscopes, not available for all, but for some they are available. And what we say is after the procedure is done, after you've used that sheath, the scope itself needs to be thoroughly cleaned and disinfected or sterilized according to the regular endoscope IFU. Now, for some sheaths, there may be recommended alternative processing instructions, and we say follow the IFU for both the endoscope and the sheath manufacturer to know what to do with them, and contact the manufacturers if there's conflict so that you get validation information and just more data on what you want to do with those. And then we do within ST91 have a section on what to do if you have IFUs that conflict. So that would be our recommendation for that. And last slide for me before I pitch it back to Garland is quality control. And this is a topic I feel very, very strongly about. We have had many quality parameters built into the sterilization process for sterile processing for years and years and years. We have not historically had that for endoscope processing. Now, all we had traditionally was the test strips that show that the disinfectant is above the minimum recommended or minimum effective concentration. But other than that, what else did we have? So now with ST91, and we see a lot more engineering of quality parameters in there. So we have done our cleaning. We're testing that we've done our cleaning by using a cleaning verification test. We're inspecting the scope externally, internally. We're still doing all of that stuff for our AER, but we're making sure we're doing all the preventative maintenance on it. We're changing filters. We're doing our self-disinfection or waterline disinfections. We're putting things on preventative maintenance. We're testing our leak testers. We may be testing the cleaning process in our AER itself. So we need to get to the point where we're demonstrating control of our process, because we know things flowed out of control sometimes, and then that leads to poor outcome for the scopes and ultimately our patients. So engineering in these quality control parameters, although it is going to take more time and cost more money to process endoscopes, we are really upping our process quality and therefore directly impacting our patients and improving their outcomes. So as far as quality control goes, follow all the written IFUs for every type of equipment that you're using and engineering in those the guidelines that we have in st91. So looking at verification and monitoring of personnel performance, work practices, adherence to guidelines. Somebody should evaluate the process. There are recommendations out there to audit your process. Infection prevention should be doing that for not only sterile processing, but endoscope processing, looking at all aspects of that. Put together this multidisciplinary team to make sure you have comprehensive written quality assurance and safety program for all aspects of endoscope process. And we do include details within st91 about what all should be in that written quality assurance and safety program for endoscopy. Again, we don't have time to go through all those details today, but so important to make sure we have these high quality practices in from the start. I'm going to turn it back over to you, Garland.
All right. Slide. All right. So talking about product recall and outbreaks and reports therein, in our policies, we have to have in our policies what steps we take when an item is recalled. It has to be in there because you never know when an event may happen. You never know when a recall may happen from either the endoscope, the equipment, or even the chemical manufacturer. And we need to know, okay, if this does happen, what does our policy say? Who do we need to contact? Infection prevention and risk management should report any outbreaks to local and state departments, and the facility as a whole should report these to the FDA. See, this is where we talked earlier about the MOD database, which we'll get into that slide shortly. But the device manufacturer needs to be notified, the equipment manufacturer, and the chemical manufacturer. This is how we get those reports in our facilities about a recall. And so they'll spell out exactly what we need to do and let them know if we have this particular product or if we use this particular product. Because there have been situations where there's a recall out there, but we may not have this particular scope or this piece of equipment anymore, and the information wasn't updated. So we need to have this in our policies. Slide. Device repair. It's important to inspect the endoscope for defects and malfunctions during processing and prior to patient use. It was talked about earlier when you're doing your leak testing, right? When you're doing your inspection, when we are manipulating the control knobs on those scopes, that is where we tell if the angulation is off. This is where we should know how these scopes are supposed to operate. You should know if you have a stiff angulation. You can tell if the scope was used in a procedure and the locks was on the knobs, and you can tell if it snapped. So when you are in a situation like that, okay, we send this scope out for repair. The angulation is too tight or it's snapped. You don't want to put that back in service. You want to clearly identify those defects. All of that should be included in the policy. We should also have preventative maintenance as part of our policy. How often should whoever we have performing the PMs on our equipment? That should be in there. How often are they coming out? Are they sticking to that? And staff should also be aware of that as well. It's not just a leadership role. Staff should know, okay, these scopes were processed or PMs were done on these machines at this time, or the scopes at this time. It's time for PMs. Next slide. Loan scopes. Now, this also should be part of our policy. Even if we get a loan scope from a manufacturer, from the OEM, it's important for us to still inspect that incoming scope. I've had a situation where I received a loan scope that came broke. The angulation was damaged and it failed leak. So it's important to make sure that you inspect it just as you would a scope that's in your facility, even though it's loaned from the manufacturer or a third party. You want to contact the loan company to report that issue. Not saying that they will blame you, but you want to make sure that they understand how you received it from them. You want to reprocess all loan instruments or scopes before you use them and before you return them. So we can't just have the thought of it came from the vendor, it's fine, we'll use it. No, we don't do that. It's just like loaned instruments in sterile processing. What do? You go ahead and you process them all over again, and then you put them in service. Slide. New product evaluation. I'm not sure how involved folks are on this meeting with product evaluation, but this is very important to establish a multidisciplinary committee. The information that should be collected is this device that you're bringing in FDA cleared, and where is that information? Research on it. There should always be research on something that you're bringing in, because you need to know exactly what you're getting. Literature and IFU, expert opinions, report from peers that may have been trialing the products. All too often, we may get items in our facility that no one told us about, and we can't process it because why? The IFUs does not allow for the chemical that we have, or they don't allow for use in the particular AER that we have. So it's important to make sure that if you're not on there and you play a part in the equipment that comes in, make sure that you're part of this multidisciplinary team. Sometimes we think that it's just senior leadership or it's just those that we never see, but we should definitely be a part of it, because as processors, we know what we have in our area, and we would like to read the IFUs, and it's important to read the IFUs before a product comes in. So here's this references and citations for this mod, the FDA mod database. If you have a moment, please write this down if you don't already have this information. But this link is accessible as well. It'll be available with the slides. But you can look here and you can see product problem, product class, event type, model number. You can find a lot of information on devices that you may not have thought were considered high risk, as we mentioned earlier. We have an amazing appendices, new and updated appendices, keeping cool, basically the environment and how comfortable an environment is, or how it can be, and sometimes how things change drastically as far as how hot it is in a room. Purchase consideration for AERs, repairs. Here we go with the IFU conflict management, right? What should you do if your scope says something, then your sink? If your sink says something, then your detergent. If your detergent says something, then your AER says something different than your scope cabinet. And everything says something different than aami. So this is in there on what steps we can take when we see that conflict. Visual inspection, cleaning verification, as we talked about throughout the document. Simethicone. Not sure if anybody's had this conversation, but this was a big topic that was discussed, and the use of simethicone during cases. That is discussed in this update. The use of high-level disinfectants and or sterilants. Storage risk assessment. Again, there's many, I'm quite sure everyone from the 159 participants on the call have different hang times that they hold their scopes before they use them. It talks about the storage risk assessment and what that looks like when determining how long you will store your scopes. Drying, that's also in there, and what that means and what that looks like. Now talking about the TIR, I know that there was a question at the beginning. We talked about it. We expect it to be complete by this year. We actually have a meeting next week where we will discuss it, and hopefully we want to have it done by this year, but if not, early next year. It's an amazing document. We've been working on it for a while. We were working on it while we were trying to complete 91, so we were double dipping. And this covers TEE probes, vaginal probes, rectal probes, and dilators. It is going to be an amazing document for both users and users. It's a great document. Slide. So Here's the information on how you can actually get a copy of this 91 document if you have not already purchased it. Your facility should be a member of aami. If they are, you can find out what your membership number is so you can get your membership price for the document, and you can purchase it right there at that link. References, st91, TIR99, mod, and it's noted on the slides. Here's our contact information for both Marianne and myself. We appreciate you taking this 2 hours out of your busy day to listen to us talk and to give you these updates. Hopefully it was helpful. As was mentioned throughout the presentation, there's a ton of questions here, and we are going to compile all of these all of these questions, and basically have it as a frequently asked questions follow-up. That way, all of these questions can be addressed in detail, and hopefully you can have the satisfying answers that you need. Marianne?
Yeah, thanks, Garland. And thank you to everybody for taking this whole 2 hours out. I'm sorry that we couldn't get to all of your questions. It was really interactive, and I appreciate that because now I'll know, what to dig through in the document. Garland and I will look through and try to nail down some of these answers for you. I did see a couple times we got a question about compiling our answers into a document and sending that to all attendees. I'll definitely check with aami on that and see if that's something we can do. If not, we can answer that individually, but we'll try to put together some of those questions into a subsequent webinar, and that's one of our options to do it. But I know that an FAQ would be appreciated. I'm seeing a lot of chat about that, so that's great. What the boil down here is it's a whole new document, innumerable amount of changes in it, no timeline associated with it. I get that question a lot, but I recommend you start, right? Get new st91 as soon as possible and start moving through it and really creating that crosswalk of non-compliance issues. Where are you at currently in line with this new guideline, and put an implementation strategy in place and hold people accountable to due dates and all of that, because there is a lot in there, and we owe it to our patients to really up the quality of our processing. So you guys know this is something I'm extremely passionate about, and I want to see us make it a safer practice for our patients, and this is a great way to do that. So I appreciate all the support over the years, and I kept telling people we're close. So I'm just so relieved we're finally there. And thank you, Garland. You did a wonderful job today.
Thank you.
All right, everybody, have a great day. Appreciate it. Bye-bye.
Thank you, everybody. Thanks, Marianne and Garland. That was an awesome presentation. As our presenters mentioned, there are a lot of questions. What we'll do is we'll pull it, get those answered, and we can post that up in the learning management system for everybody once they're answered. We'll give our presenters a few days to get those answered, obviously, because there's quite a bit. So once it is posted in the LMS, we'll reach out to everybody to let you know. Again, this was recorded and will be made available in the learning management system within the next 24 hours. Somebody from our education team will post that and then email the group to let you know when it's ready for viewing. But again, thank you for your participation, and please do keep an eye out for future st91 training opportunities. We're going to be working with Marianne and Garland to piece together some other training programs and get that out to you all. So thank you very much. Enjoy the rest of your day and take care.